Marker-assisted technology, DNA extraction protocol. Take leaf samples from rice plant of 0.2 molar grams collected from glass houses and fields. We take the finely cut leaf sample in mini spotter plates placed in alternate wells. Now we take CTAB for extraction buffer. CTAB is preheated and it comprises of 5 moles of NaCl, sodium chloride, 1 mole of tris, 0.5 moles of EDTA, 1% of PVP, 2% of CTAB. This process is for protein denaturation and phenol removal. Of CTAB is mixed with the sample and grinded finely. After grinding, we again add 350 μl grams of CTAB. In the next step, we take 700 μl grams of sample, adding equal volumes of chloroform, adding isoanyl alcohol in ratio 24:1, which forms vortex. After getting this sample, we take it for centrifuge process with 10,000 rotation per minute (rpm) for 15 minutes. After first centrifuge process is done, we get our supernatant, whose equal volumes are treated with chilled isopropanol. Then this solution is kept to incubate at room temperature for 3 to 4 hours or for 1 hour at 4 degrees Celsius. Once the incubation is done, we take it again for centrifuge with same RPM, but this time for 10 minutes. After centrifuge is done, we discard the supernatant and collect it in pellet. We add 70% of ethanol to it in next step. Then we tap the tube gently and centrifuge it for the last time at same rate, but for 5 minutes this time, and we discard it with ethanol after centrifuge is done. We then have to dry the pellet overnight. In the next step, we add solution of IXTE, which comprises of tris plus EDTA. The tubes are then kept for incubation at room temperature for half a day to properly mix and dilute the DNA. Once we have done it, we go for the next step, that is PCR, polymeric chain reaction, to multiply the DNA strands.